talks. These talks are largely focused at the cellular level. This is going to be one of the more intense basic science talks, so are you ready? We're gonna see if we can get through this. So, as many of you probably know, the DNA within each mammalian cell, or our genetic material, undergoes a striking degree of compaction to pack some 1.7 meters of DNA into just a 5-micron nucleus that you can't even see. Now, in this elegant and highly coordinated process of compaction, the DNA, here the individual base pairs, is compacted some 5,000-fold into this complex tertiary structure called chromatin. Now, chromatin is just a fancy word for our DNA wrapped around protein so that it can tightly condense and compact all our genetic material. Now, as you can imagine, the reason we have this is because this is a very important organizational scaffold for our genetic material. But the compaction itself also poses very significant barriers to critical cellular processes, such as transcription, turning genes on at the right times, etc. And so our cell has developed several ways to deal with this. There are several processes, I'll list them for completion: DNA methylation, histone modifications. These are just names of modifications that can go on the genome to regulate expression. And for the purpose of my talk today, ATP-dependent chromatin remodeling. It sounds like a mouthful. So what are ATP-dependent? These are energy-consuming complexes. And how do these regulate chromatin? So this is, meet the BAF complex. This is the BAF complex. This is a large 2-megadalton complex, which actually sits upon your DNA and rolls down DNA, thereby moving these compact nucleosome structures and opening up DNA so that it can be transcribed and certain genes can be turned on in a timely and appropriate manner. These are large complexes, as you can see from this diagram. There are multiple subunits within this complex, and it rolls along DNA in every single one of our cells in our billions and billions of cells. And as you can see, forget the nomenclature on this slide, but I show this only to show that over the course of evolution, these complexes, which exist, have largely gained, lost, and shuffled subunits in response to evolution. So these complexes have occurred in yeast, Drosophila, flies, and now in mammals. And as you can see, yes, we have evolved, it turns out, since flies and yeast, and we have a greater genome size, and so there are more complexity within these complexes. So our group studies this, our entire lab studies these complexes. Now, certain things we've learned over the course of studying these complexes. It turns out that the different combinatorial assemblies, we all studied combinatorics, different combinatorial assemblies and constellations of subunits come together to drive divergent sulfates. In fact, just by switching the subunits of this complex, we can turn a fibroblast into a neuron, etc. So this complex, for example, gives rise to embryonic stem cells. These complexes are hallmark to neural progenitors, or those cells that give rise to your cells in the brain, these cells in cardiac progenitors. And so the take-home message of this is that the different combinatorial assembly of these very interesting protein complexes drive a great deal of biologic specificity. And when I joined the lab at Stanford that studies this and really discovered all of these proteins, I was really interested in cancer, but this lab did development. So why did I join this lab? As you can see here, this is a cross-section of a developing brain. So what happens at the very last mitotic exit, as neural progenitors, or the cells that are going to give rise to the rest of our brain matter, undergo their last division and become post-mitotic mature neurons, there's a dramatic switch. Again, the subunit switching of this complex actually drives development. So this was a very intriguing example. This is a cross-section of the brain, very important role in neurogenesis, development of the nervous system. And it turns out that these complexes play critical roles in the development of a host of tissues, from the brain, as I mentioned, to the formation of cardiac chambers within the heart, to muscles, to thymic development. So suffice it to say, this is very important. The knockout of any gene encoding one of the subunits of this complex results in embryonic lethality, so organisms can't develop. And we've learned several rules. We take all these genes, all the products of those genes have to be assembled into a 15-subunit complex, and there's just certain rules as to how they come together. There's several hundred possible combinations. We need this for biologic diversity. They're very stable. This is just a way we look at stability. And there's certain positions that can only be occupied by one gene or another. So, now here comes the exciting part. Why do we care about these complexes? So over the course of the past several years or so, thanks to next-generation sequencing, there has been a surge of exon and whole genome sequencing studies in human cancer. And these have repeatedly revealed frequent mutations to the genes encoding the various subunits of this large chromatin regulator. And in a recent analysis, taking into consideration all of the subunits that we know are part of this complex, we can estimate that these mutations are present in greater than 20% of human cancers. That's quite substantial. And so we can see here that these mutations, if you take all the genes again and look across a panel of different tumors, all the tumors really that have been sequenced to date, this is already out of date because there are now over 100 sequencing studies. This was at the time there were only 44 or so. But the point of this slide is that you can see that these genes are mutated across a wide spectrum of cancers. So they're broadly mutated, much like P53 or other very important tumor suppressors you may have heard about. Yet, if we're really trying to prove that these complexes, when perturbed, can play a driving role in cancer and therefore should be a target, we would need to say that that's the only event that really, and the founding event that drove that cancer. Yet these mutations often occur in the context of other mutations. So how, as a biologist, were we able to really prove that these play driving roles? It turns out, in some very specific forms of cancer, which turn out to be more rare forms of cancer, there are specific, genomically well-defined driving events that we know give rise to this tumor. We know if we put a certain mutation or a certain translocation in cells, we can actually develop tumors and drive tumors. So in this set of biology that I'll show you, we took a rare tumor, a tumor that I had never heard of when I wanted to study this process, called synovial sarcoma. It's a very aggressive soft tissue malignancy. It's about 10% of soft tissue sarcomas. And we used that to understand a great deal about this process called chromatin remodeling, and vice versa. We took chromatin remodeling, what we studied in the lab, to really uncover the mechanism of this horrible disease which spans both pediatric and adult patients. So what is this? So in 100% of patients that have this malignancy called synovial sarcoma, 100% of them have one genetic problem. And that genetic problem is a translocation. Turns out that this gene on chromosome 18 called SS18, I didn't know what it was when we first started studying it, as well as SSX on the X chromosome, come together to create a fusion gene. So jumping genes, essentially. One gene went to the other, and now it's a fusion gene. And at the protein level, genes make protein, so at the protein level, we see that we get this fusion protein as opposed to the wild-type protein. So what did we discover? We showed that in 100% of patients that have this, we were able to collect cells from these patients and interrogate this biologic mechanism. We showed that this malignant fusion protein, which drives these cancers, absolutely usurps the structure of these ATP-dependent chromatin remodeling complexes. And thereby, by physically changing the structure of these complexes, it ejects and prevents the assembly of these other wild-type normal components. Those components get degraded. Excitingly, this is the gene fusion, here's the protein fusion, it integrates into the complex. This complex, now when it rolls down the DNA, causes for completely different pathways to be turned on, which are actually pro-cancer. Now, excitingly for us, we showed that this was actually reversible. These patients still have one normal copy of the normal protein that's supposed to be in these complexes. We showed it was reversible, and if we could reassemble these complexes, the cells would arrest, so you get death of these synovial sarcoma cells, and this presents a very open avenue for therapeutic discovery. So our whole lab takes advantage of cancer genome sequencing, which is just in this exponential phase now, and takes these findings, we're the recipient of these findings, and tries to uncover their biochemical and functional consequences. These sequencing studies have brought about an unparalleled level of information which we can now use to try to identify biologic targets, therapeutic targets, as well as develop personalized medicine. So can we extend now what we did in a very rare tumor type, synovial sarcoma, which many of you have never heard about, can we extend this now to more common cancers such as lung cancers and breast cancers, colon cancers, kidney cancers, etc. So can we use the principles we've learned here? So this brings about several questions. We care about the structure of this complex, what's bound to it. Are there additional avenues for targeting the various components bound to this? Understanding the architecture. We've drawn this complex. You've seen about 10 renditions of this complex, and that actually reflects the fact that we actually don't know where these puzzle pieces actually go together. There is no structural information for this. So this really begs understanding of the architecture, how these complexes come together, as well as structural information. So what I've told you today is that these genes encoding these subunits are very frequently mutated in human malignancies across a wide range of human cancers. Very specific subunits of this complex are mutated in specific cancers, suggesting that they play very important tumor suppressor roles. The discoveries I showed you in synovial sarcoma are the first to really demonstrate that altering the complex composition and structure can directly impinge on our gene expression programs, and we're asking the question, can we now identify therapeutic avenues to reset faulty complexes as a roadmap to treating these cancers? Thank you.